Bismillahirrahmanirrahim. The new topic of our discussion is prediction of the transmembrane domains in a protein. Although there exist several tools on the internet regarding the prediction of the transmembrane domains in different proteins, one among them is the TOPCONS. It predicts basically the transmembrane domain based on the conserved and the consensus sequence that has already been identified in literature. So it's the web interface on the home page of the TOPCONS web-based tool that is used for the prediction of the transmembrane domains and the signal peptide in a particular protein. So for example, we have this protein ZMPSTE24 and we want to predict whether this protein contains any transmembrane domains or not. We will copy this sequence and we paste here in the query box of this tool. Make sure that the sequence format should be in the FASTA. Then you have the option, for example, you want to set any name for this job. This is optional, but for example, you may write the name of the gene. And the email is only required, for example, if you have a number of different proteins. But as for example, we have only used a single sequence for the prediction of transmembrane domains. So here we have the option to give any email ID, or even if we don't give, it doesn't matter. So for example, we will here suggest, we will here write the email ID, and then you just need to click on the submit button. So here within a few seconds, it depends, if the sequence length is small, as in our case the protein length is only 475 amino acids, so in that cases you will get the result in less time. And for example, if you increase the protein size or if you add more and more protein, then definitely the analysis will take longer than the usual. So here you go for the result. You can see here initially all the preliminary information, that is sort of information you have given. Then before the start of the result, you can see here this one is the key to all the predicted result. For example, this window basically indicates your protein prediction. Here you can see your protein prediction, but before that you need to understand that which color and which line uh, account for which prediction basically. So this red line indicates inside mean that it's the cytoplasmic side of the protein. Blue indicate outside means the extracellular side of the protein. This gray indicates the transmembrane helices from in to out. It means from cytoplasm to extracellular. Unfilled box indicate the transmembrane from out to in, and the solid black color indicate the signal peptide. So just scroll down and just come to the exact prediction predicted by the TOPCONS. You can see here that uh, this number indicate the position of the amino acid. We start from 1 and it goes up to, for example, here 450 and beyond even. So you can see here that initial few amino acids, they are basically outside the cytoplasm, inside the cytoplasm. Here you can see, sorry, outside. Then transmembrane outside to inside. So if you see this like pattern, a pattern like this one, that inside, then for example outside, transmembrane helices from in to out, from out to in. So it indicates that yes, this protein contains a transmembrane domains. But if you don't see this like pattern, then it indicates that the protein is not a transmembrane.